The homeobox containing genes encode transcription factor. This we have already discussed in the previous lecture. In early 1980, Walter Gehring, Gehring, and his associate William McGinnis, Ginnis, and Michael Levin, Michael Levine. So in early 1980, Walter Gehring and his associates William McGinnis and Michael Levin, working in Switzerland, and also another group of scientists working in University of Indiana. They were Thomas Kaufman, Kaufman, and Matthew Scott, Matthew Scott, Scott. So these two group of scientists, one group working in Switzerland and the other group working in the University of Indiana, they were studying the antennapedia complex. As already discussed, that Hox gene, they have 8 genes. And what are those 8 genes? One is the cluster of genes designated by antennapedia complex. This antennapedia complex has 5 genes, as I have already mentioned earlier, and the bithorax gene, it has 3 genes. So they were studying the antennapedia complex using a recombinant DNA technology. Now these investigators set out to isolate and clone the antennapedia gene. This gene is a member of antennapedia complex, as I have already mentioned. So they had isolated the antennapedia gene, which is a member of antennapedia complex, from Drosophila, and as a part of their study, they prepared a DNA that could hybridize to the antennapedia gene. Now they were greatly surprised to see that the antennapedia gene or the DNA hybridized with both the antennapedia gene and the nearby segmentation gene. The antennapedia, one of the segmentation gene is Fuji-Tarazu gene, Fushi-Tarazu. So I have already told in previous class that in Drosophila axis specification there are a certain set of genes. The first set of genes are called the maternal genes, which specifies the anterior and posterior region in Drosophila. Then there are the segment gene, and these segment genes have pair rule genes and other genes. So to their surprise, they saw that the synthesized DNA, which was supposed to hybridize with the antennapedia gene, also hybridized with the segmentation gene Fuji-Tarazu. The antennapedia gene and Fuji-Tarazu gene must have had some DNA sequence of close similarity, because part of each gene hybridized with the genes of antennapedia gene as well as the segment gene. Further hybridization studies demonstrated that this particular shared stretch of DNA also was found in bicoid gene of the Drosophila gene, bithorax complex. In some other part of Drosophila genome and in genes in other insect species. So this was really intriguing, and the studies revealed that in most bilaterian animals, I have already told you what bilaterian animals are. Those animals which can be, when cut vertically, can be, we get the right side and the left side, and we get a mirror half of the animal. So in most bilateral animal, the Hox genes are expressed in staggered domain along the head to tail axis of the embryo. And these findings were obtained by doing the hybridization studies, when certain stretch of DNA was prepared, and this certain stretch of DNA not only hybridized with the Antennapedia gene, but it also hybridized with the segment gene, bithorax genes, that is the ultrabithorax, abdominal A, B. So this piece of DNA, which was synthesized, hybridized with so many other genes, this suggested that all the genes, Antennapedia gene, segment gene, that is the Fujitarazu gene, bicoid gene, Antennapedia gene, all these genes must have certain sequence which is common to all of them. So this important sequence was found out to be 180 base pair, and this particular 180 base pair is present in a homeobox gene. So there is a homeobox gene which make the homeobox protein, and within the homeobox gene is a sequence of 180 base pair, and this sequence of 180 base pair is called the homeobox, and the protein which is synthesized by the homeobox gene is called the homeobox protein, and sequence specific to 180 base pair yields 60 amino acid, and this 180 base paired translated protein is called the homeodomain protein. This I have already shown in the last lecture. So we can say that this sequence of 180 base pair of DNA is called the homeobox, has been shown to be part of many genes in many animal and plants. This conserved sequence ensures the structural organization in all the animals and plant. That is, where the anterior region will be, where the posterior region will be. The homeotic gene of animal contains the homeobox, but the organ identity genes of this homeobox, we can say, is present in only in the animals. For plants, there are similar but other gene. Mice and humans, they have been widely studied mammal. So mice and humans have cluster of homeobox containing genes. So the homeobox containing gene is called the homeobox sequence gene.